Hi, good morning. Here we are back at manganese and yet another finding that came out about 2 weeks ago on the ability of manganese to be oxidized as fuel for a bacteria that was found by a scientist at Caltech. I've got the reference in the notes here. This has been going on for a long time. This has been going on since the beginnings of time, and let's take a look at our embryogenesis situation where the fertilized egg in the first 0 to 16 weeks, the mitochondria are hanging out in diploid shells during neurogenesis. Basically, it's the formation of the brain, which happens under a thick embryonic membrane, like 6-8 times thicker than it turns out to be later in the fetal development. And the biogenetic needs are such that the woman becomes anemic, low iron, low oxygen. Doctors, please reconsider giving iron to first trimester. Anemia is part of the process, as I have found. This is from studies with Northeast Brazil in the microcephaly and their needs during first trimester that do not include iron ore mining tailings, excess iron or manganese. Only after the first trimester, first 16 weeks, does iron and manganese come in and flesh out these mitochondria. These are the mitochondria. They're just hanging out for 16 weeks. Here in the embryonic development, the fetal, oh gosh, I should be able to draw this. The brain stem, the telencephalon, mesencephalon, all the parts of the brain are differentiating, including the brain stem. That happens in this period. All of that happens, goes from a single cell that has been fertilized, the male sperm's mitochondria fall away or disintegrate, and the female mitochondria stays and creates these, while this is happening, diploid shells, mitochondrial shells. They're empty. There's nothing in them yet because the floodgates have not opened for their fleshing out. They wait until the embryogenesis occurs. And what's operative in this arena? Zinc. Zinc is very highly reactive. We know that. It becomes highly toxic later on. But in embryogenesis, to get that egg differentiating until that trillion neuronal cells, I don't know how many exactly come in that first 16 weeks, but a lot, maybe most. It's pretty astounding. Zinc and copper, right? Copper is our friend in embryogenesis. The virus doesn't like copper, does it? In fact, everything that happens in embryogenesis is what seems to kill the virus. That's the dichotomous counterintuitivity of how this virus how we have flipped our biological environment, and the virus is moving in on that. Almost like, you can almost predict it by thinking, well, what's the opposite of what we need is what this virus is taking. And we remember we evolved in highly oxygen, well water, spring water, groundwater, rain, of course, but not the purified stuff we have now. We'll talk about that in a minute. And, food was hard to find. We had to wait for trees to bloom. We had to wait for mushrooms to pop up. We had to wait for Thanksgiving turkey to walk by. And now we have flipped that. We're eating all the time. Processed foods, it's pretty obvious. And then our water is so purified, but it's not necessarily healthy. Not as healthy as it could be, because this is evolved water. This is processed. This is, 2020 water. Let's call it that. And then the oxygen levels, we are stressed. We're not out there exploring and foraging and waiting and pacing and stalking food or sniffing it out with our olfactory that, is not only a deep breath. It's like, what is in that? Can I eat it? What is that? Is it friendly or can I eat it? Basically. So we have flipped. This is 2020 BC, and this is 2020 AC, or AD. Oh, okay. Okay, so there we have it. And be careful, my dog is very protective of me up here. He just has no idea what this whiteboard's going to be doing to me next. And you don't either, huh? No, it's good. So 0 to 16 weeks, zinc and copper. Have we got that one? And when you put zinc and copper on carbon, what do you get? A bright white LED light. That was in one of the earlier sessions. That is something. And so when you see the egg differentiating, you see it's like, oh, it's like it's not even fireworks. It's just like bright white light coming out of that. That's what you see on the differentiating fertilized egg in the presence of copper and zinc. And so then, this is the first 8 weeks. And then when you go to 8 to 16, these things kind of flip. Copper becomes more operative and zinc becomes less operative. So their proportions switch out. And there's a research on that. There's papers on that. Not enough, but enough to support what I'm saying here, theoretically. Okay, and then afterwards, and there's a very thick membrane here, very thick. It's like ultra thick. You're not getting no oxygen needed. No, we don't want. It's very anemic, hypoxic. It's a very protected environment, and while these mitochondria hang out in shells, so the iron levels go down. I'm not sure what manganese is doing at that point, but it's holding back. It's got nothing to work with. I'm not sure what it's doing then, but I know that iron has been sequestered because, and who knows, manganese may be storing up here. I don't know. So then in the second and third two to infinity, there's a gradual curve where we go up, we become more and more oxygenated. By the time we're 30, all that matters is oxygen. All that matters for our health is oxygen. All that matters is staying oxygenated and doing the bada bada boom. Functionality, and deactivating that amygdala. You need that amygdala when you're a little rat running around the forest, or brat, whatever you are, whatever you term, whatever. Kids, running around. I don't know. You got to think back to 500,000 years ago. Whew. Kids had to be pretty, quick and nimble, and that is a hypoxic activity. Do that on the piano. If I have a really hard thing to play, I hold my breath. Then it happens. Then I have to tank up again. So and basically what happens to this zinc, it gets locked away. I'm trying to put a lock here. That's a lock. It gets locked away, and the mitochondria keep it locked away. And a copper is used on the heels of calcium to protect the integrity of cell activity. And it's also controlled by the mitochondria. Okay. And so, and all these 1 trillion neurons turn into, I think, 100 billion. So 10%. 10% of these, of what happens over here, more or less, survives to become our, DNA expressed, carved, cleaved. This is what we need in this environment, right? And if you grew up in Barbados or somewhere, you will have a whole different physiologic set, pace, rhythm, etc., music memory, survival set, as it were. Okay, so so we see in COVID-19. Let's go down to COVID-19 here. Oh gosh, it's hard to make this transition, but it's not impossible. And I've done it in many of the earlier. I've attempted to look very carefully at that transition. How did this thing evolve? Where did it come from? What is it doing? How does it have the power that it has over us? Well, flipping the planet is one thing. We have filtered out manganese. Manganese is a problem in modern filtrated water. Why? It manifests, and that's what this paper is that I wanted to talk to you about, that bacteria with a metal diet is discovered. Well, in dirty glassware. That bacteria with a metal diet is called Gallionella, and it clogs up water filtration plants because of the manganese. So what do? They filter out the manganese. Manganese also will dirty up the water, give it a brown color. And iron ore mining knows this. And if you put manganese and iron, which is what they call the tailings, the leftover sludge from iron ore mining, turns the water red. Well, how do they cloak that? How do they hide that? You'll see in most iron ore mines, they have a little teeny budget line, $10,000, $12,000 for aluminum. And aluminum will clear it up. Glug, 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 microcephaly. Because iron and manganese do not belong here. Iron and manganese do not belong in first trimester. No manganese. Manganese is my mitochondria purple. And iron is my oxygenated, iron cleaves to oxygen. None of that in here. We want zinc and we want copper and, other yet to be discovered biosubstrates, perhaps. It's pretty simple, though. So in fact, I did an experiment when I started taking manganese about 4 months ago, however long this has been going on, maybe 5. And I had zinc tablets and I had manganese and I had copper pennies. And so I was very interested in the zinc and copper and the zinc and manganese and the And so I had these little plastic bags, Sealy bags, and I put water in each of them. And I put manganese and copper. I forget exactly what I did. I have to repeat it because I put them in a dark closet and I totally forgot about them. Like this bacteria with a metal diet. This guy went off for a few months and totally forgot that he left this bacteria with some manganese. And when he came back, it was black and it had created substance. Let me read a little bit of this for you. Very, very interesting. Nodules, let's see, relevance. Okay, no, manganese is all over the seabed floor and like grapefruit type balls, lots of deposits of them. So biomass, chemosynthesis. Yes, that's what manganese does, especially at surface level with algae. There's a chemosynthesis where it moves in and I believe it's part of the tangles that we find in Alzheimer's plaques. In fact, I've followed those tangles and you look at normal aging like this, right? Well, you first start with A beta is produced, and you're looking at hypoxia. You're looking at all the risk factors that cause hypoxia, and A beta is actually cleaved in the mitochondria. Does that surprise anyone? I think we've talked about that. Mitochondria, oxygen is, A beta is cleaved in the mitochondria. And when it's hypoxic, it's going to cleave the A beta 42. Maybe it's 42. And so then, okay, so hypoxia, we've worn it out. We've worn out the hypoxic mechanisms, and manganese is starting to shoot out to rescue the mitochondria. And at which point, what comes next? Dehydration. Because when manganese cleaves that, the water molecule is gone. And so we're looking at tau. Now, this is an example of how when manganese plus H2O, as they're saying in this paper, creates what do they call it? Filaments, I believe they call it. Filaments. All right. Yields energy. I mean, it's basically what happens at the bottom of the ocean when there's sludge and no oxygen and big grapefruit-sized balls of manganese, groundwater. Manganese oxides take the form of a dark, clumpy substance. But yeah, they talk about filaments here, or bear with me. If you look at Gallionella, you'll see what I mean. They're tangly things. Tangles. There you have it. I mean, so dehydration, manganese, tangles. Low oxygen, mitochondrial distress, A beta. So all roads point to manganese. That's that there. Now let's go back to COVID, if we can. And I'm kind of excited about this manga photopure. There's another one that's out that is manganese. Manganese chloride doesn't seem to work. I don't think that's a good one. That doesn't sound right. I haven't looked that up yet, but it does. I don't think it's been approved. And there's another one that's it's an MM something. I mean, it doesn't seem to have it has a different conformation. But what I like about manga photopure, is very interesting, is that it has a lot of oxygen, phosphate, more oxygen all over the place. Goes in, let's see, has a nitrogen. I mean, nitrogen seems to, these nitrogen terminals or N terminals, I guess that's nitrogen. I'm working on it. Surrounds the manganese, and there's more oxygen. So what does manganese do? It gets oxygen. And so, what's it going to do if it's already got oxygen surrounding it? It's not going to, it's stabilized, because that's what it does, is get the oxygen. And then there are other, there's plenty of oxygen terminals on this, right? And this is mirrored on the other side. This is only half of it. You got to flip it for the other side. So it's really quite an escort to get manganese into these brain areas. I hope Dr. Stevens is watching and listening, because of all the people, I know you can understand this, and you will make the claim, if this is the claim to make. And maybe these vaccines are more of a metallurgic type vaccine, or include a metallurgic component. Because we know now, I've been talking about this for days, that manganese is in the center of this virus. This virus cannot function without glycoprotein. Yes, we know we have given all the material where it can have all the glycoprotein and just multiply by the gajillions, right? But right in the middle are manganese ions, two little doobie-doos that like literally cleave the viral RNA, the 3-unit, and the viral RNA, the leader viral RNA 5-unit. And also contributes to making this double membrane vesicle DMV in or through the mitochondria. Now, how does a mitochondria make a double membrane vesicle? Well, that is the study that just came out. It's validated by another scientist. I can tell you what I did what I found when I put my little bags in and brought them out later. I'm like, and I also put, I threw in some biomass, like cactus leaf or something. So I may have confounded that. But in one of them, it was like this skin-like substance. I'm like, now that's creepy. It was a distinctly skin-like substance, and I think it was with the manganese, the copper, and the zinc with the biomass. Creepy, but that's what I found. That's what I got, and I didn't publish on it. I'm just too busy doing all the busy things that I do. But that's good. And talking to you, I'd rather talk to you than sit around slave away at a paper. Put it on film, baby. Okay, and where are these made? These are made in the mitochondria. The leader RNA goes into the mitochondria. It may get its zinc there. I don't know if it comes in with the manganese or gets it when it comes in. Let's look into that. Let's just look into that, shall we? Where does it get that manganese? Because it's a non-specific protein. It's NSP15, and it has has manganese in it, and the UENDO, I think, that actually cleaves the DNA. And the leader one, from my understanding, it comes from the mitochondria. The leader. No leader, no virus. And the double membrane vesicles come from the mitochondria. So, as far as I know, mitochondria don't make anything inside of themselves, but they do signal for things to be made, like this manganese energizing bacteria makes stuff. It creates stuff. I know in the bottom of the ocean floor, when manganese has, these 3 metals, it creates sediments. Oh my goodness. Okay, as we're looking at this, let's just keep talking about it. So what's happening to the mitochondria in the virus? Big fat filled with electrons. Okay, how does the mitochondria usually work? It uses, the electrons are coming and going and coming and going. They're coming and going and coming and going. And that's how we get our energy to keep us alive. Well, all those electrons are being corralled. And so how are you going to turn manganese 2, our friend, into manganese 3 that is at the bottom of the ocean? That's where you're going to find the manganese 3, and that creates sediments. Oh boy! Well, you add an electron. You add an electron. Oy, oy, oy. Plenty of electrons in the big fat stuffed COVID-19 mitochondria. All right. So what's going to keep this from happening is my I beg doctors to please try to give mitochondria so that the manganese 2 is coming out and creating the antioxidants. So also get more mitochondria, more manganese. I do make mistakes and sometimes how I say I need to correct it on some of the films. So here's the COVID-19. It just needs 2 little ones, 2 little manganese to make it go, get into the mitochondria, create the leader viral RNA, to create the double membrane vesicles, and maybe it spits them out or signals for them. I'm not sure, but from my understanding, that's where the actual house for these things come out. And we provide all the glycoproteins because we've flipped our planet and we're halfway there anyway. The second viral RNA comes from the nucleolus, which is in the nucleus, which is in the cell itself, and then the endoplasmic reticulum creates, I don't know, maybe it does the rest of these glycoproteins, while the mitochondria and the leader RNA create the vesicles themselves, and it all gets created, takes over the cell that way. Takes over the cell. This is our cell. So what increased manganese does, what I'm saying is here, just a little bit of manganese and little guy, and little guy, quick little log, very delicate little dainty little log on this virus, right? Well, when there's too much manganese, when you add manganese, it's like you're not doing anything. The virus does not get created. That's pretty clear. So to increase the manganese is the most logical, obvious approach. No, the microcephaly infants, it's the opposite. They needed purified water. They needed purified water. But they got it was all laced with iron and manganese, which is not welcome in embryogenesis. And if you look at these kids, they're kind of super chunky, hunky Neanderthal, big old ears, big, super, they're kind of super fleshed. So, that iron is supposed to be buried. That manganese is supposed to be in the groundwater. Iron is embedded in rock. Keep it there. So is petroleum, buried. Keep it there. Manganese is flaky. It's it diffuses into the water, and we need it. Okay, so I guess that's where I'm at with it all. And there was just a paper that just came out on this bacteria that can have a metal diet. All right, that's Origins of Life, Harold Morowitz, under the auspice of Dr. Jim Olds and my great, awesome Krasnow colleagues and friends, that where I studied many aspects of this, thermal vent, deep sea thermal vents, where origins of life are suspected to happen. Now, this is a perfect example. There's no oxygen down there. That's the phenomena of this bacteria, that there's a bacteria. Oh no, I put it in like mitochondria color, but it's bacteria. And mitochondria, Gallionella, this is nothing new, really. I mean, he found it in the lab. That's great. I found, I found tissue-like when I played with the metals, combinations of them. It was copper, zinc, copper in Tri-1, and then I put a biomass. It was a cactus leaf, I think, and manganese. I think that was the combination. although I had several of them, but I was just amazed that this material had come about. And there was some dark too, just like he found. Okay, so let's see, this bacteria way down in the bottom of the sea, way down in the bottom of the sea. We're gonna add a little bit of manganese and see what we will see. 'Cause down in the bottom of the sea, there's no oxygen for you or me. Just water and a thermal vent and a bit of manganese. Way down in the bottom of the sea. So, okay, I'm being silly. But so this bacteria, most bacteria, I guess, like oxygen, but there's no oxygen. But what it does have is the manganese and the dynamic manganese 2. Or is it? And I know that the triple electron manganese does interact with sediments down there. So it's all, this is great science. The microcephaly in Northeast Brazil, unfortunately, great science. And so this COVID-19 is also science. So thank you for listening to me today. I guess I've gone on a bit. A lot of information and some new science on how manganese, a bacteria with a metal diet, first bacteria found to use manganese as their source of fuel. It's the first science that's found it and published on it. But it's this type of bacteria has been around for a long time. In fact, if you look at Gallionella, that is a bacteria. So environmental Jared Ledbetter, environmental microbiology, Caltech, postdoctoral scholar Hang Yu, published in Nature. Wonderful aspect of microbes that they can metabolize unlikely materials like metals, yielding energy useful to the cell. Well, that energy, that manganese energy is certainly useful to COVID-19. It certainly is. Why? Because COVID-19, it's in the middle of it, doesn't belong, you're not part of it. It's just the energy to unlock this thing and to cleave. That's what it does. So it gets our DNA, and this manganese does bump. It cleaves it, and it does bump, and it cleaves our DNA to pair up with the RNA, cleaves our RNA to cleave up with the RNA of the virus and make virus. Please tell me I'm wrong. Somebody please tell me I'm wrong. Because where there's causation, I just keep finding more and more evidence and support, scientific support. It's not even evidence, it's support of this theory that manganese is going to play a big role in helping us with this virus and teaching us lessons about how we need to honor and protect our own mitochondria in the most solemn and humble way, and also honor and protect the elements of this earth that have enabled us to evolve to the degree we have. I don't think happy hypoxia is anything to write home about. In fact, that is the matrix. That would be the matrix. We just have a scaffold shell that is just fed and fueled by flash images. Boom, 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 boom. What are we? The frogs sitting there watching life go by? No. We're much deeper and have phenomenal potential. Oxygenation will do that. Healthy mitochondria will absolutely one and only do that. We know that. Scientists know that. So that's our next frontier. And we are still evolving, aren't we now? Thank you for listening. Thank you so much. I'm so grateful to have my colleagues here every day and to be able to give these lectures during this pandemic. And I'm off to the American Psychological Association conference, and I hope you can join me. tomorrow, Friday at 3 p.m., where I launch my lecture film premiere on music as medicine to help the worst case Alzheimer's normalize, hyper-normalize, come back to life, and talk as though they didn't have the disease. Those are deep paleo-limbic, subconscious, involuntary processes that we all have. We micromanage them to death. And once that micromanagement area dies, then, those limbic paleo involuntary processes become stronger again, and music has the power to resurrect them. We're in such a great learning curve here. Unfortunately, disease teaches us so many things if we let it. So let's put metals and manganese into that triage vaccine. Mangaphotopure. Thank you. And safety and health and blessings to everyone. Have a good day.